In this lesson series entitled Creating a Memory, From Encoding to Storage to Retrieval, we will follow the course of events in the production of a memory stored in the human brain. But before we tell that story, we'll need some background information. So we'll be covering these concepts very quickly in this introductory part 1 of the lesson series, and then we'll tell the story of how a memory is formed in future lessons. So we're familiar then with the surface of the brain called the cerebral cortex. And in fact, we should be familiar that different regions of the cerebral cortex appear to be engaged in different functions. They have different jobs. Now, how did scientists make this map of the brain? 3 lines of evidence, basically. The results of brain damage. What happens if a part of the brain is damaged? What happens if a part of the brain is stimulated with electrodes? And if we record from a certain part of the brain, under what circumstances will that part of the brain become active? Those 3 lines of evidence gave scientists the ability to sort of map out the cerebral cortex. And if you kind of take a look at what's going on in the back half of the brain compared to the front half of the brain, we can sort of roughly think that the back part of the cortex is answering the question, what is happening? What am I seeing? What am I hearing? What am I feeling on my skin? Is there language in my environment, etc.? And the front half of the brain is more about, what should I do? Should I say something? Should I move my body? Should I continue thinking? Should I make a decision? Should I use my feelings and emotions to help me make a decision? So this is a rough approximation of what the 2 halves of the cerebral cortex are doing. We also learned about a short-term memory system. We'll call it working memory. And this system allows us to hold information and manipulate that information over the time course of seconds. And this is a very important capacity. We'll associate this with a sort of a short-term memory system. We're going to put the box up here in the frontal cortex next to the word thought and forethought. So, when we're having a verbal thought, that would be the content of our working memory system. But in fact, scientists have found that there's other areas of the brain, the parietal cortex, that are also engaged when we're doing any task that involves working memory. But we're going to put the box in the front part of the brain. So whatever's in our working memory system is what we are consciously aware of. And it may well be that we're just in the environment, observing the environment. So all the back part of the brain here, what we're seeing and feeling and hearing, all of that information can be sent up and loaded up into the working memory system, and then we will have the conscious experience of seeing things and hearing things, etc. The working memory system is really important because the contents of the working memory system can then be sent back to other areas of the cortex for more processing. And so we get this ongoing loop of activity. The brain sends information up into the working memory system, and then the working memory system sends information back to those brain areas for further processing. For example, if we're having a verbal thought, that thought that we're having right now can activate the language areas, and then we have the next thought in our chain of thinking, because the activity in the language areas then would be loaded back up into the working memory system. Or the thought that we're having now can be used to search our long-term memory system, and we can retrieve a memory that is related to what we were just thinking about. When we retrieve the memory, that memory will be loaded up into the working memory system, and we'll have the conscious experience of remembering something. So it's a very important system in the brain. Now, we know brains are built out of microscopic living cells called neurons. In this picture, we see a neuron up here, and we see a neuron down here in the bottom left, and we see that the top neuron is generating an electrical signal that is racing down the axon of the neuron. The electrical signal goes to all of the terminals at synapse sites, and when the electrical signal gets to the terminal, chemicals are released, as you see in this picture here. Chemicals are released called neurotransmitters, and these chemicals are like keys. They bind to these receptor proteins and they unlock them, as it were. A channel will open up and charged particles called ions will rush into the target cell, and that creates an electrical signal in the target cell. Here we see a neuron, again the cell body. We have the branching structures are called dendrites, and this provides more surface area for lots of synapses from other brain cells. Here we have 2 synapses here, and another 2 here, and 2 here. And the diagram is indicating that there is activity at these synapses. So chemicals are being released, the neurotransmitters are released, and that's having an effect on the target cell. Small electrical signal is being generated at those synapse locations. Notice this synapse here is not currently active. So an electrical signal did not arrive at the terminal. No chemicals being released. Now what neurons do is they add up the activity of all the synapses, and if the activity is above a threshold value, then the cell body is going to generate an output electrical signal that will race down the axon to the terminal and cause the release of neurotransmitters to affect the next brain cell in the chain here. Because these brain cells are living cells, they can grow, and this is how we can learn and remember things. So this feature of brain cells to change the strengths of connections at the synapses is called plasticity. And because of neuron plasticity, our brain literally changes its structure as we learn. And those changes are the basis then of memories. Let's take a look at the details here. Here we have a synapse, and imagine this is a synapse prior to a learning event. So there's some baseline strength of this synaptic connection here. And here we see a little bit of the details of how the communication works at the synapse. Electrical signal arrives at the terminal. Neurotransmitters are released into this gap. There is fluid surrounding all of these brain cells. So these neurotransmitters diffuse across this gap. Right, they make their way, and they're going to be bouncing into the membrane. If they latch onto the receptor in just the right way, they will open a channel, and then other charged particles, like sodium ions, for example, will rush in to the target cell through these channels, and that is an electrical signal. The movement of charged particles is an electrical signal. So electricity is the movement of charged particles. In a wire, it would be electrons moving around. In brains, it's charged ions moving across membranes. So the effect, then, of the electrical signal arriving at the axon terminal is that it generates an electrical signal in the target cell. Now, what happens after learning? Well, remember, these are living cells. They can respond to events in their environment. What's going to happen here, immediately after learning, the target cell is going to insert some extra channels in the membrane of the spine. Remember, this is spine, a little outgrowth of the dendrite. And if you increase the number of channels here, then for a given number of neurotransmitters, you can allow more charged particles across the membrane and therefore create a larger electrical signal. So right away, a short-term effect of the learning that just happened is that the target cell can increase the number of receptors, and that has the effect of strengthening the communication between those 2 neurons. But what about the long-term effects? Scientists have evidence of several events that are occurring that are thought to be the basis of long-term memories. And one is a growth process. The spine itself grows bigger. If you have a bigger spine, you have more membrane here to insert more of these receptor proteins. And so these can be maintained for long periods of time. And so again, that's going to strengthen the communication at this synapse. With more receptors, there's going to be a larger electrical signal here. So we've got a larger spine, more receptors, and in addition, it may well be that for each electrical signal that arrives at the axon terminal, more neurotransmitters are released. So with more chemical keys, as it were, the chances of opening up these locks, these channels, increases. So here we have 3 sorts of biological changes that are happening when we learn, and a long-lasting change, of course, then would be the basis of long-lasting memories. But we're not going to worry about the details at the synapse for most of the rest of this lesson series. We're really just going to use a simple icon to represent that learning had some kind of effect on the synapse, had strengthened the synapse. And we're just going to draw, right here's a little synapse prior to learning. And then after learning, the synapse is stronger. And we'll show that by just having a larger kind of terminal here. What we're not showing are more receptors. We're not even showing dendrites here. So we're not showing receptors and so on and spines. But we're just going to let this be the icon for neuroplasticity, the growth process that occurs after learning. The synapses get stronger. All right, we also learned about two kinds of memory: episodic memories and semantic memories. And by episodic memories, these are the kinds of memories that are very detailed. They're vivid, have lots of detail, and they might have the context of when and where you were when this event happened. So we'll call that the circumstances of learning. So if you might be in a classroom, you're using the microscopes, you're feeling the microscope, you're observing things in the microscope, maybe it was Halloween, you're wearing a costume. If you can kind of recall all those details and the where and the when, the context of the learning situation, then that's an episodic memory. By contrast, semantic memories are more about sort of just the information that was learned. Semantic memories do not have the added extra detail of the circumstances of learning. It's just, what was it that was learned? And we use different words to refer to these kinds of memories. For episodic memories, we say things like, I remember, or I can relive that episode, right? We have the feeling of transporting ourselves back in time and with the richness of the details and the context. Whereas for semantic memories, we say things like, I know something. I know about amoeba and paramecia, but I really don't remember when and where I learned it. So semantic memory is all the stuff we know about the world, but we don't remember the circumstances of learning. And so there's an important relationship between these kinds of memory. After all the stuff that we learn about the world, at one point it started as an episode of learning. So the contents of your episodic memory are going to be used to install new information in your semantic memory system. But as I said a moment ago, oftentimes what happens is we forget the context of the learning situation and the details of the event itself, but we hang on to what it is we learned, but we forget the circumstances of learning. So there's an important relationship between these two kinds of memory systems. Episodic memories provide the contents for what we eventually will store in semantic memory. And it seems that the brain is perfectly okay with forgetting the context or the circumstances of learning, so long as we install some rich knowledge about our world. So what would it look like in our brain to have knowledge, say, about tigers? Well, given the back of the brain here has the sensory systems, scientists think that these perception areas, sensory perception areas, are also the locations where brains are storing important aspects of memory for different things in the world. So, for example, the sight of a tiger, what a tiger looks like, would be stored. The memory for what a tiger looks like would be stored in the visual areas. The memory for what a tiger sounds like, the roar, would be stored in the auditory areas. The memory of what a tiger feels like would be stored in the touch areas of the brain. We have a memory of the sound of the word tiger. And if we were literate, we would have a memory of the sight of the word, the letter string. Now, what's going on in the brain then is the brain is linking all of these separate memory traces, linking them together to form what we would call the concept of tiger in the brain. And this is kind of a useful thing for brains to do, because then if you see a tiger, you can then activate what a tiger sounds like. Or if you hear a tiger, you can activate what a tiger looks like. So notice then that the memory, the semantic memory of what it is to understand what a tiger is distributed around the brain. There's no one place in the brain that is the tiger memory place. The sight of a tiger is stored in one area. The sound of a tiger is stored in another area. The word sound is stored in yet another one. But the brain is going to link all of those tiger bits of information together to form the concept of tiger in our semantic memory. Now, to build that semantic memory, we are now going to introduce a key structure that is necessary to increase the information stored in semantic memory, and that's the hippocampus. The word memory here is written on the bottom of the temporal cortex here. I think they put it there because underneath the temporal cortex is a really important structure, the hippocampus, and it's on both sides, so we have two of them, the left and the right. The hippocampus is critical for converting a learning episode into knowledge stored in semantic memory. Here we see a sheep brain that has been dissected. The left or the right temporal cortex has been dissected away, and there's the hippocampus right there. And here we've pulled out the hippocampi of several sheep brains to display them. Now, we know that the hippocampus is a critically important structure because of the conditions of neurological patients. So patient HM and Clive both had their hippocampi damaged for different reasons. HM, he had surgery to treat epilepsy, and Clive had a viral infection. But for both individuals, the hippocampus on both sides was not working, and as a consequence, they could not make new memories of their life. They couldn't make new episodic memories, and because episodic memories provide the content for semantic memory, they couldn't make any new semantic memories either. So they couldn't learn about their events in their own life, they couldn't remember the events of their own life, and neither could they then learn about things in the world, because you need to be able to make an episodic memory to provide the content for what we're going to store in semantic memory. So by studying patients like HM and Clive, scientists came to the conclusion that the communication between the hippocampus and the cortex is critically important for building long-term cortical memories. So the hippocampus has to be able to talk to the cortex, communicate with the cortex to give us the long-term memories that then we will be able to retrieve for a lifetime. This process is called consolidation. So we're kind of converting newly learned information into long-term memories stored in the cortex, but we need a functioning hippocampus for that to happen. Sometimes this is called system consolidation because we're talking about different brain areas working together. Or sometimes it's called memory consolidation. But the idea is this process is creating long-term memories in cortical brain areas. In fact, scientists have found that this process of memory consolidation, a lot of it is taking place during sleep. So when the hippocampus, during the day when we're learning, the hippocampus is going to be making new memory traces of the new stuff that we're learning. And then at night, what appears to be going on is the hippocampus will reactivate those new memories, and they will communicate with the cortex to strengthen the cortical memory traces for that same information. And again, this is happening at night when we're asleep. Now, let's change it up and think about the learning process from a psychologist's point of view. And here, you get like a flow diagram. So there's no brains shown in this diagram, but just the flow of information, in a sense, through two kinds of memory systems in our brain. So here we're in classroom, we're listening to a lesson. Our working memory system is going to be processing the information of the lesson. And if our hippocampus is doing its job, the hippocampus will help us then, over time, make a long-term memory of the information from the lesson. And that's the consolidation. So if we were successful in making long-term memories of the information, then we'll be able to retrieve that information at a later time. So now comes the test, and we read the test question. We load up the test question in our working memory. Now the working memory, its job is to search. So the contents of the working memory will be able to search our long-term memory. And if we studied well, we will be able to access that information and retrieve it. So the retrieved information gets loaded up into the working memory system, and then we will answer the test question. Finally, we'll wrap this summary up with a couple of analogies that we'll encounter in the lesson. First, we'll take the computer analogy for memory. And like brains, computers have two kinds of memory, a sort of a short-term, a fast-acting memory. It's kind of like our working memory, and a slower but larger capacity long-term memory system, the hard drive. So we can think of the functions in a computer. When you enter data into the computer, it can write the data into the long-term memory system, the hard drive. And then we can access that information. We will read the data when we need to access the information stored on the computer. So we have a write function and a read function. And this is how sort of the short-term and the long-term memory system interacts in a computer. Well, in brains, we have learned that the hippocampus is critical to do the writing function. When we learn something new, the hippocampus has to be functioning so it can help the brain write the newly learned information to the hard drive, the cerebral cortex. And we learned that neurological patients like HM and Clive, they have a dysfunction. Without a working hippocampus, they can't do the writing function. So they have short-term memory in the sense that you can have a conversation with them and whatnot, but anything that happens to them, they're not going to be able to make a long-term memory of that episode because they've lost the part of the brain that does the writing to the hard drive, the hippocampus. Now, there is a weakness with this computer analogy. After all, in computers, all information is stored with the same strength, and that's not true for brains. So, if you might know that in computers, it's a binary language. You use ones and zeros to kind of store information. Chains of ones and zeros can represent information. There is no sense in which one piece of information is stored more strongly or weakly than some other bit of information on the hard drive. But that's what it is in the brain, right? So since neurons show plasticities, memories can be weak or strong depending on how much growth happened at that synapse. And so as learners then, we do want to pay attention to this fact that it may not be enough to just learn something once. We need to give our brain a chance to really strengthen the connections between the relevant neurons so that we can hang on to that information for long periods of time. So since the computer analogy does have a weakness, I'm going to shift the analogy to a printing analogy for memory. So here we have a printer, and the idea is the hippocampus is going to help print memories in the cortex, the long-term storage system. So if the printing process, if we're not using much ink, then we're going to have sort of a weak memory stored in the cortex. With a greater amount of ink, right, more ink applied to the paper, the image will be richer and brighter and more vivid, and we'll have a stronger memory. If we use a lot of ink, we'll have a very strong memory. So if we want good, strong, long-term memories, we have to help the hippocampus do some good printing up in the cortex there. So that's our analogy here. The hippocampus is a printer, and if we want long-term memories, we need to help the hippocampus either make a strong printing the first time or study repeatedly so that the hippocampus can keep adding ink and really strengthen those cortical memory traces. There's another analogy that we will hear in the lesson series, and that is that the hippocampus is a teacher. In fact, before it becomes a teacher, it's a fast learner. So this is a school analogy. So the hippocampus is a fast learner. What that means is the hippocampus has a lot of plasticity, has high plasticity, so it can make really strong memory traces after one learning event. And then later, during sleep, as it turns out, the hippocampus is going to be activating those memory traces and signaling the cortex to strengthen those cortical memory traces. And that's sort of like a teaching function, and we call this process consolidation. And we'll see that a lot of this consolidation process happens during sleep, a fairly recent discovery. So that's our background then for this lesson series. We've covered a little bit about brains and neurons and working memory, neuron plasticity, episodic semantic memory, patients like H.M. and Clive, and this idea of the hippocampus being critical in the process of memory consolidation. In the next part then, we will begin the journey for how a human memory is made.